Hello and welcome everybody to the Future Tech Week. My name is Eric Prem. I'm the director of Tumor and a partner in the FedFX project organizing the Future Tech Week events in collaboration with the European Innovation Council pilot. The Future Tech Week 2020 fosters communication around visionary, high-risk and long-term impact research and emerging technological trends funded by the European Commission under EIC Pathfinder FET as a driver for future breakthrough innovation. The Future Tech Week in this year is an online event to celebrate the EIC Pathfinder FET program. Now, the FET program has been around for over 3 decades, and I think it has gained a reputation for its leading-edge research focus, but it is also a program that focuses on longer-term technology developments in many areas of science and technology. And as such, it is a highly ambitious and competitive program funded by the European Commission. It is therefore a particular honor to welcome Professor Moser to his special talk for the Future Tech Week. In 2014, Professor Moser and colleagues received the Nobel Prize for Physiology and Medicine for their work on grid cells. And I'm excited that Professor Moser will be giving us a short introduction in his work, especially because Professor Moser was also involved in a FET project in a leading role. And the project focused on furthering our understanding of grid cells and also investigated how these newly discovered neurophysiological principles could be used for developing new technologies, for example, for robot navigation. Now, I hope this was an appropriate introduction in the work of Professor Moser, so I'm looking forward to your own short explanation, Professor Moser, for the next 20 minutes or so, and then we'll have a short question and answer session. Thank you, Professor Moser.
Very much. So I appreciate very much the introduction. I think it was fairly accurate. So what I will do then, as you indicated, is to share with you some experiences from the FET project that I was involved in. So let me just share the screen now. And now you should see the file. So here, this is this is my 10, 15, 20-minute summary of a project that took place from 2013 to 2017 under the funding or under the scheme of the FET Proactive Grid Map project that it was called. So, okay. So, the participant of this project, it was 4 groups. So first of then I was the coordinator working at the Norwegian University of Science and Technology. I worked together with 3 other groups. One was a neuroscience group from the University of Edinburgh, headed by Richard Morris. And then in addition, we had a computational neuroscience group from CISA in Italy, from International School of Advanced Studies. And finally, a technology group from the Technical University of Munich in Germany. And these 3 groups then spanning the range from basic science on the brain to technological innovation, headed towards trying at least to learn from the brain and extract information from the brain that could possibly be used to actually improve computers and particularly robots. So let me begin with some background. So the starting point was the invention of computers, and computers have been around for at least 80 years now. But although they have totally dominated our own field or everyday work of everyone in the world, it is still the case, or at least was certainly the case at the time when we started, that computers are still extremely good at speed and power. So computers are excellent for performing precisely pre-instructed operations on large sets of data, and doing this extremely rapidly, exceeding what humans can do completely. But it's still the case, and was definitely the case when we started the project, that computers still had some challenges when it came to doing many different things and tasks at the same time, and especially when the tasks were ambiguous. And this is the case just in everyday situations that we as humans and all animals experience all the time. Lots of things is going on in the environment around us. We have to focus our attention, but still process many channels, what different people do at the same time as I'm speaking. For example, planning the next steps of my talk, and also being distracted of whatever I hear from the neighboring room at this time, and doing all of this at the same time at very high speed. So computers haven't been so good at handling this in parallel and interactively until quite recently. And for that reason, one of the aims of the project was then to try to find out what is actually the brain doing, what are the computations that brains are doing that actually help us do this so fast and so efficiently and with so little error. So we wanted to learn from the brain, the nature's most advanced computer, and see if we could extract any information that possibly could be used to develop better computers in the future. And then particularly we focused on the cortex, which is the outer sheet of the cerebrum, which is the main part of the brain. And this outer sheet is then shown in gray around here, is there when where we think that most of the intellectual activity takes place. So let's then focus on one of the functions that takes place in the cortex of the human brain, and that is the ability to find one's way. So the brain's spatial mapping system. It is about how to find one's way, how to know where you are, and how to get from there to other places. So very simple in a sense. We do this all the time. Both humans and all animals are completely dependent on doing this in order to survive, in order to find food, in order to find a mate, and so on. But still, it is only very recently that we have learned about this system. And in fact, we have learned more about this system than many or even most other high-level brain functions of humans. So what you see here is a human brain. You see the temporal lobe on the side here, and in red, this is shows an area called the hippocampus. It's actually inside the brain, so it's sort of folded into the brain and into the cortex. And then next to it, you have another area shown here in blue, which is called the entorhinal cortex. And those are two important parts of the cortex that I want to focus on now. And the reason is that these two areas are so important for the navigation system, and in particular for helping us to map our own location. And why focus on this? Well, for this project, the whole reason is that here we do at least understand some of the underlying computations that take place, and we thought that we might perhaps extract some of those to the benefit of technological innovations. However, this is a human brain, and that's a very complicated structure. So we think that we can actually benefit from learning or extracting information from animal brains, and then in particular rats and mice. So there's a long history of studies in rats and mice, and particularly when it comes to navigation or finding one's way, which these animals are extremely good at. And using rats and mice as model species has actually taught us the most of what we know about space and navigation in the brain. So we focused on rats, and it is possible in rats, has been done for about 50 years, to actually record activity from their brains as they are performing behaviors. So this shows a rat, or it could also be a mouse, running around in a small box where it can localize small pieces of chocolate, which they like, and that makes them run around all the time. And at the same time, it is possible to plug a small device, small sensors, to a connector on their brain so that we can record from activity in their brains while they are through a cable here. It can also be wireless to a computer, and then it's possible to pick up the electrical signals that underlie the activity of the entorhinal cortex and the hippocampus as the rat is running around. So what's going on in cells in this brain area as the rat is finding its way in this box. So we recorded then from the entorhinal cortex of rats and mice. So in 2005, and then we made our biggest breakthrough probably, and which also was used as a motivation for the Nobel Prize in 2014, and namely the discovery of grid cells. So in the entorhinal cortex, we found that there are cells that have localized spatial firing, and that fire in a periodic hexagonal pattern. So what do I mean by that? Well, let's now consider this rat or mouse that's running around in this box. And then let's look at down here, you have a picture from an overhead camera that shows in gray the trace of the rat as it's running around in the box over a period of half an hour. So it's almost visited every possible place. And then in black, each black dot shows a position where one particular cell was active. It means that that cell fired an action potential, as we call it, which sent an impulse to other cells in the brain. And you can see that the activity of this particular cell in the entorhinal cortex was not random. It occurred when the rat was at particular places, and those places formed a very regular periodic pattern. So you can see it appeared in dots, clusters, and those clusters formed a hexagonal pattern that spread all the way over the recording box. And I can show that in a different example here. So what you see here is a different cell, but now I put red lines on top of it. And you can see that this is just to illustrate that it's a triangular or hexagonal pattern that repeats itself all over the surface of the environment. So this is a grid that covers the entire environment. Each cell shows a grid that covers the environment, and for that reason we call these cells grid cells. And because these cells contain a metric information about distance and direction, and is based on the animals running around in the environment, then this in many ways serves as an internal GPS of the brain. And although we don't really call it a GPS, I mean, that's very often used as an analogy when people try to explain the workings of these cells to a broader public. So that was the background. That was the situation when we decided that we wanted to apply for this project. And then why did we apply? Well, it was because these grid cells, they are among the first non-sensory cognitive high-end functions that the brain has that have been understood in some detail mechanistically. So we wanted to exploit this window into a high-level area of the brain, and then ask if this could be used to actually improve computers, and in particular computers that navigate in the environment. And the aims are then shown down here. So it's a three-step objective for the project. First, we wanted to determine the mechanisms by which space is represented. So how does this brain area work, and especially in realistic settings, which was necessary for in order to bridge the gap over to technological applications such as robots. Then we wanted to use these insights to infer the computational algorithms that this brain. Then we wanted to use these insights to infer the computational algorithms that this brain area uses. And then finally, with the help of having these computational algorithms, we then wanted to see if they could be applied to instruments like computers, and in particular robotic agents that navigate around in the world. So could we help machines to navigate using the principles that the brain has invented millions of years ago? And here are some of the outcomes of the projects at a broad level. So first of all, we did during these years find out a lot about the mechanisms of this grid pattern. We found out how the pattern is wired and what causes this hexagonal pattern to be created. I'm not going into the details of that because that requires a lot more time, but we found that a lot can be explained by the interactions between many cells in this brain area, the way they work together, and the hexagonal pattern of firing in many ways is an equilibrium state that the neural circuit arrives at under certain conditions. And then this hexagonal map is then updated as animals move around in the environment and reflects the animal's position, and can be used to infer actually where in the environment the animal is. So we extended, as I said, this to more realistic environments than just running in a box. We made complicated boxes with different rooms and walls, and try to simulate at least environments that are more similar to indoor environments where some robots move around, and then of course ultimately environments out in nature. And we did that by adding additional features, borders, walls, blockades. And so how does this affect the grid cells? How do they operate in such more realistic situations that we need to understand if we want to extend this to a robotic system? And then at the next step, we also saw that this actually happened several places in this entorhinal cortex area. There were different circuits that computed space or location at the same time on their own, and operated at least to some extent independently in a modular way. So a classical example of parallel processing that might also be interesting for the development of next generation computers. And then importantly, we established and developed models to try to explain what was going on, because these explanations are necessary if we are to bridge the gap over to applications. We need the rules or the laws or the computations that we can apply in a machine in order to see if they can operate in the same way. And we did that. We, together with our partner at the Technical University in Munich, we developed a test arena for robotic experiments. He developed robots that moved around on the floor in his lab, and trying to do so and to find places based on rules that were actually developed from what we thought we had extracted from the mouse or rat brains as they moved around. And finally, I think perhaps the most important here is that the development of proof of principle demonstrations that such algorithms can actually be exploited to create technical innovations that in some ways can be better or hopefully be much better than the applications that are the state of the art. And then let me just finish by summing up what I think is a general impact of this project. So first of all, it continued to make a huge influence on neuroscience, and particularly I think it helped us understand two things. First, how is this grid pattern created? So it's a major computation of the brain, and one that could be quantified and could be effectively modeled. And we also managed to try to adapt these insights to more realistic environments where you bounce into things and have to make detours and shortcuts, and try to understand how could the brain actually take these things into account in order to compute the most efficient path to a goal. And in that process, then I think actually the most important element of the project was the contribution to the growth of computational neuroscience. So computational neuroscience is a branch of neuroscience that has become increasingly important because it tries to describe the operations of the brain at an algorithmic level. What are the rules, the laws, the computations that take place among neurons that actually result in a certain function, such as the creation of an internal updated map. And then having these algorithms available, then we at least took some steps towards using those for creating technology, robotic implementations that navigated around in the environment, and also use some of these parallel or modular computations that we saw taking place in the grid cell system of rats and mice, and we know also happen in the human brain. So I think this bridge from basic neuroscience through computational neuroscience towards application, this is perhaps, it was the goal of the project, and is also where I think the project left a contribution. Of course, it's a long way to go because the brain is still a big mystery. We only have a few windows into understanding its underlying computations. But I mean, using the insights from the brain can, I think, in many ways be a stepping stone towards creating better machines, intelligent robots. And it's not always the case that the brain has the best solution, but quite often evolution has found ways that we could learn from as we create tomorrow's computer. And that's the long-term benefit for society, for sure, I would think. So I think with that, I'll just thank you for the attention. It was a brief presentation, but hopefully you got at least the gist of the project. Thank you. Now I'll just stop sharing then, and then we are back at the with both of us in the mainframe.
Thank you so much, Professor Moser. Fascinating work, and thanks also for the let's say relatively lightweight introduction into neuroscience. I can tell you that from a computer science background, this is all very fascinating. Computer scientists tend to be hugely impressed with studying brains because of this. From a computer science perspective, so much is going on in parallel. I mean, everything's going on in parallel. Yes. There's this massive firing of neurons. And I would actually like to say that a GPS is fairly simple compared to what's going on in the brain, because the problem is in a computer you have a single processor that follows instructions. You can write down numbers, but how do we achieve the same function in this large network of of neurons that are interconnected is far from trivial, even mathematically. It's not easy to see how the brain does these wonderful things.
No, it's
And the
still
second
amazing.
issue,
And it's also
yeah,
not only that these things go in parallel in many systems, but they interact all the time. That's a really, really complicated thing. So it's all dependent on each other.
yes, it's a very good point. And computer scientists always try to limit these interactions. You know, we always write functions with very clear interfaces,
Yes.
so as not to have any of those interactions in
Yes.
our opposite of what you're trying to do here. But on the other hand, when we look into nature, all living systems, or at least animals, all they do is they move bodies through environments. So it is to be expected that a large part of the brain, and indeed very old systems of the brain, do exactly this, and they probably do it in a very good and robust manner. They navigate bodies through space.
Yeah.
So, and if you think that we still do not fully understand this, it's not surprising that our robots are still a little bit brittle and far from being robust.
Yeah, no, it's still a way to go, and I think at the moment that's our challenge. I mean, you can create robots that solve this in completely different ways than the brain, just using a sensor that then recognizes what you see on the image, similar to the retina. But I think you will still then face these challenges of taking so many things into account, and so I think. It is, I mean, we can learn a lot from robots. I think starting with a GPS is a place to start because it is so simple, as you say, and then, but hopefully then by revealing these algorithms of the brain that can actually be used for much more complicated processes in the end, and especially when we understand these parallel computations and also the interactions between many systems.
But let me ask you one question then, maybe a level higher. So, I mean, science is all about understanding nature, and technology is very much about building things. Yeah. And innovation goes one step further. It's about selling things. Yes. So what's your take on this? How important is this this characteristics also of the FET projects, where you're trying to really combine the two? Is this a good way of doing research?
I think basic science and technology are totally interdependent. So, and it goes both ways. So first of all, I think technological development is always dependent on knowledge about how things work. And I think we see this just now in the era of the pandemic. I mean, developing a vaccine so fast can be done because we know how the immune system works, we know what a virus is like, and we can create a vaccine that operates on the right targets of a virus. And all this happened almost within weeks in the beginning, just designing the appropriate target. And this is because there's basic knowledge about the human body and about viral agents, and it's the same for the brain or anything else that we need this basic knowledge, then we can apply it. But it also goes the other way around, because a major limitation for example, neuroscience and many sciences is the availability of technology. So the brain is so small, it's so many computational units spread over the entire brain that picking up that little activity has always been a challenge, and there new technologies are the driving force. So technology also influences science again, but I think it's a bidirectional relationship. And what I liked about the FET program is that you have to think far ahead and not only take the next steps, and that is what these projects do. They don't always lead to major breakthroughs within a 3 or 4 year funding period, but just getting people on that track to look far forward, that is really what is important for development. And that's not only for science, that's also for technological development.
I believe this is a very important point. So you're saying it is indeed, like FET does, worth looking a little bit further ahead even in technology development, not everything, although we're still interested in innovation, but it's not around the corner. It's about some more fundamental changes here.
Yeah, that's absolutely true.
Okay. Now, let me close this meeting with a more personal question. How has being a Nobel laureate changed your life? I mean, I'm grateful you found the time, but we all imagine, you know, a Nobel laureate being only invited to talks and not being able to do any more research. It's a way to kick out some.
Yeah, you hit the point there, because when you get the Nobel Prize, then your life changes in the sense that you become some sort of ambassador for science too. You speak out about things and people listen to you. But I mean, these things, not only going to talks, but also doing science policy and fighting for science funding, that all takes a lot of time. So that, and it's an important job, but I also try to balance it still very actively towards maintaining a lab, and a lab that is at the forefront of the field still, and having lots of good people in it. And if I want to have these lots of good people working in the lab, they will only come if the lab still is at the international forefront. So it is a difficult balance. I'm fighting with that all the time, but still think that doing my science, finding out how the brain works, is still my highest priority.
And I hope you are still able to squeeze in a little bit of technology as well, and not just science.
Oh, definitely. So we're now at a point where there's actually another technological revolution taking place in the brain sciences, which is that until quite recently, people, and including myself, we recorded a few cells at the same time. That's all we could do. But now you can have thousands of cells at the same time. And why is that important? Well, it's because all these complex functions like finding your way and thinking, planning, memory, that's all about interactions between large numbers of cells that work together. And if we are to understand that, we have to monitor the activity of all of them and play with them, turn some on, turn others off. And you can't really do that until you have a totally different technology. That technology, with the probes for recording many, many cells, and even trying to perturb them in certain ways, that is now becoming available. And for that reason, I think there's another revolution now going on, which brings neuroscience to the neural population level, where you look at thousands of cells at the same time.
Brilliant. Many thanks. I think, Professor Moser, this was a brilliant way to kick off the Future Tech Week. And I think it demonstrates how important this relation between science, technology, and innovation can be, and how important it is having programs such as PhET, also in the future when it's moving over to EIC as Pathfinder.
Yeah. I cross my fingers for that and hope that these programs will not only continue to exist, but they will get higher priority. So good luck with the Future Tech Week.
Thank you so much.
Hi, I'm Silvia Scaglione. I'm the coordinator of the B2B project. Within the B2B From Breast to Bone project, we are developing technologies for supporting biomedical research, adopting recent advantages in fluid tissue culture system and 3D bioprinting. B2B was inspired by the difficulties and experimental limitations that the biomedical
among large groups of cells define the architecture and function of the tissue, depending on where it is in the body.
In a research line called FIT Proactive.
In EVA Nano project, we are using
evaluate the efficiency of any given nanoparticle design,
where we will
People.
Of the result. Organs. But the three features play an essential role in cancer development and its spontaneous formation of metastases involving the passage from one tumor to another fluidically connected organ. On the other hand, in animal models, the human-derived cancer is surrounded by a non-human physiological system, which of course can impair the reliability of the result. To overcome this, we want to develop an in vitro alternative, more clinically relevant than the available in vitro models, and that is specifically conceived to study the spontaneous metastatic process.
The B2B device hosts two organoids connected by a fluidic system to study the breast cancer metastasis to the bone. The process begins in the breast chamber, where cancer cells multiply while vascular cells form a micro-network of capillaries. Within this growing mass, the migration of cancer cells to the bloodstream happens spontaneously. The capillary network connects with an external macro network of bioprinted tubes covered with endothelial cells. Once they join, some tumor cells enter the synthetic tube and, following the circulation, they travel towards the bone chamber. Here, the cancer cells leave the circulation and to discover factors during their journey or hinder their migration.
Very critical in B2B. It will use macro to micro bioprinted branching and features of the blood vessels. And at the same time, they should be directly connected to the self-assembled capillaries from the tissues. And as a first application, we have selected the metastatic process of breast cancer to the bone, since it still represents a major hurdle. Breast cancer is the most common in women worldwide, and its most common metastatic site is indeed the bone. In B2B project, we will connect a patient might be applied in the future to study other types of cancer with similar features. I believe B2B will lead to significant advancement in breast metastatic cancer knowledge, finally boosting also the new drug development.
The human body is constantly affected by physical forces. When you cut yourself, cells in the surrounding tissue apply forces to heal the damaged area. In the same way, cancer cells apply forces to spread to other parts of the body. To understand these mechanical forces, we need to look at them at many different scales, from molecules to organs, and all the way up to entire organisms. Understanding how these forces work would allow us to unlock new avenues in cancer research, regenerative medicine, and the design of biomaterials to be implanted in the body. At the nanometer level, which is the level of molecules, cells stick to their surrounding extracellular matrix and to other cells using integrins and cadherins. Forces applied to these molecular links change their properties, determining how cells behave, and sometimes activating the expression of genes. Some of these, like the ones triggered by the oncogene YAP, play a role in cancer. At the micrometer scale, cells work together to respond to their environment. They combine biochemical mechanisms, such as signaling, with a biophysical response to react to mechanical forces, such as when there's an injury. Similarly, at the millimeter level, the tissue level, the collective actions and interactions among large groups of cells define the architecture and function of the tissue, depending on where it is in the body. And at the meter scale, the scale of entire organisms, all interactions at the molecular, cellular and tissue level integrate to result in organs and ultimately functional organisms. We know that forces are involved at all the different scales, but how exactly do they contribute to tissue and organ behaviour? To find out, the Mechanocontrol project will put together a toolkit to uncover cells' responses to forces at every scale. Like a well-stocked toolbox, the kit will include different tools in which the 7 Mechanocontrol partners are experts. These will range from single-molecule nanotechnology at the smallest scale to organoids and animal models at the organism scale. It's the first time that expertise in such a broad range of technologies will be put together and applied to all the different scales at which biological functions occur. In particular, Mechanocontrol will apply its toolkit to breast cancer. If the partners can understand cancer biomechanics from the single molecule to the whole organ scale, they'll be able to control mechanical forces to restore healthy cell behaviour and inhibit tumour progression. Ultimately, it's hoped that the project will lead to the development of breast cancer treatments based on the new discoveries. The principles revealed by the project's toolkit will also apply to many other biological processes and diseases.
Hello, my name is Sergio Garcia Mañez and I'm a professor of biophysics at the King's College London. My lab is interested in understanding how individual proteins equilibrate under the effect of mechanical force. And this is particularly relevant for the Mechanocontrol Consortium because we aim to understand how individual proteins involved in mechanosensing and mechanosensation in cancer cell lines unfold and refold under mechanical tension.
Hello, I'm Ana Diaz from the Leibniz Institute for New Materials, and in our lab we are developing in vitro breast cancer models that can recreate the mechanical and compositional changes of the tumor environment. Using light-responsive biomaterials, we can identify the membrane receptors involved in tumor progression, as well as the mechanical threshold in which they activate. Our studies in the Mechanocontrol project will guide the development of new therapies and new diagnostic tools for cancer.
Hi, I'm Menno de Jong from Noviocell. We are a young company based in Oslo, the Netherlands. Our role in the Mechanocontrol project is to provide our collaborators with synthetic hydrogels that cells can attach to and that dynamically stiffen as the cells that are bound to them strain the matrix. This helps our collaborators to develop new tumor models that help them better understand the mechanical changes occurring in tissues during tumor development.
My name is Pera Roca-Cusax, and I'm a group leader at the Institute for Bioengineering of Catalonia. My group, along with a group of Xavier Drapat at the Institute, study how cells sense and respond to mechanical factors from their environment, such as tissue rigidity. In the Mechanocontrol project, we study specifically how breast cells and breast tissues respond to the different mechanical properties of breast tissue and how it changes in the progression of the disease. We are applying this to understand the implications in the progression of breast cancer.
Hi, my name is Patrick Dirksen. I'm a cancer biologist from the University Medical Center in Utrecht, the Netherlands. In collaboration with Johan de Rooij, I lead a team of cancer investigation trying to understand how breast cancer metastasizes using three-dimensional tissue culture models and mouse model systems. Together with the Mechanocontrol Consortium members, we are trying to translate the discovery phase findings into the preclinical arena in order to treat breast cancer.
My name is Marino Arroyo and I am a professor at the Universitat Politècnica de Catalunya in Barcelona. In my group, we develop theoretical and computational methods to understand cell and tissue mechanobiology at a mesoscale. These methods should allow us to integrate all the scales addressed in the Mechanocontrol Consortium, from molecules to organs.
I'm Guido Giudetti, and I'm a biologist specializing in neurobiology. Here at the Sant'Anna School of Advanced Studies in Pisa, I'm involved in a neuroengineering project to improve the quality of life of patients who have had a heart transplant. The project is called New Heart and is funded by the EU's Horizon 2020 programme in a research line called FIT Proactive. What we want to do is take a transplanted heart and restore the neural connections it lost due to the operation.
A questo cuore trapiantato le connessioni neurali che vengono perse durante il trapianto. Questo è un elemento.
This is very important, since the nerves that connect the heart to the brain regulate the heartbeat. The control nerve is called vagus. So, the vagus nerve, when active, has the function of slowing the heartbeat.
Quindi è facile immaginare il fatto che un paziente che ha subito un trapianto di cuore con le metodiche attuali.
A patient who has had a conventional transplant is like a driver in a car which is stuck at 110 km an hour with no brakes.
Come se si trovasse a guidare un'automobile che va a 110 all'ora senza avere i freni. Quindi il suo cuore continua a
battere. Their heart pulse remains the same whether they're sleeping, walking or doing physical activity.
Questa situazione provoca
In the long run, this situation leads to lower quality of life as the fast-beating, uncontrollable heart eventually gets tired.
Senza nessun controllo alla fine si sfianca. Stiamo cercando di fare qualcosa che non ha mai provato a fare nessuno. Noi l'abbiamo definito un
We're trying to do something that no one has ever tried to do. We've called it a moonshot. At a certain point, we looked at each other and said, do we realize that we're trying to make Iron Man? He is a superhero who uses a special technology to help him stay alive because his heart has suffered some damage. The FIT program has given us the confidence to carry out a high-risk, high-gain research. The anatomy of the vagus nerve itself is not well known, and this is something that we will study specifically in this project. We may not achieve our aim, but firstly, we'll have developed technologies, and secondly, if the research is successful, we will literally change the paradigm of heart transplants.
Noi cambiamo il paradigma dei trapianti di cuore.
Recombinant monoclonal antibodies represent today one of the greatest therapeutic and diagnostic modalities in modern medicine, with application in the treatment of major diseases like inflammatory and autoimmune disorders, cardiovascular diseases, and cancer. More than 70 monoclonal antibodies are nowadays available on the market, with a sale which is expected to grow up to $125 billion in the next future. Traditionally, monoclonal antibodies are manufactured from batch cultures using mammalian cells, followed by extensive purification steps that rely primarily on discontinuous and cumbersome chromatographic techniques, with intermediate filtration and viral clearance. Unfortunately, several chromatographic steps are required to reduce impurities like whole cell proteins and DNA, with an increase in production costs. Crystallization is an effective, cheap, and scalable technique conventionally used to purify small drug molecules. Up to now, the purification of biomolecules directly from clarified fermentation broths has been prevented by the structural complexity and flexibility of the target molecules and the presence of impurity in cell culture media that hinder nucleation and growth steps of the crystallization process. The primary goal of the AMACRYST project is to unlock the industrial application of protein crystallization in biopharmaceutics by combining the continuous manufacturing concept with the efficient crystallization process based on membrane technology and selective recognition of complex biomolecules in multi-component solution by engineering three-dimensional nanotemplates. template-assisted membrane concept, pursued in this project, is expected to allow a selective recognition of protein molecules in fermentation room, whilst membrane will facilitate the declination step and afford crystallization efficiency. By this strategy, crystallization has the concrete potential to completely replace chromatography in the production of antibodies, with significant benefits also in their final formulation due to high biostability of the crystalline state. Amicus researchers aim to replace chromatography with a single membrane crystallization unit, developing a continuous prototype for industrial purposes, so that financial and operating costs in antibodies downstream processing will be reduced by more than 60%, thus making production costs more affordable, enhancing product shelf life, while achieving the same drug properties in terms of therapeutic activity. The Amicus Consortium involves research institutions, universities, and industrial partners from 4 European countries: the Italian National Research Council, that coordinates the project, the University of Calabria, the French Centre National de la Recherche Scientifique, l'Université Libre de Bruxelles, the Imperial College for Science and Medicine, the University of Strathclyde, GVS, Center for Process Innovation, Fujifilm Dirksen Biotechnologies.
Sono Silvestro Micera, professore di bioengineering.
I'm Silvestro Micera, Professor of Bioengineering at the Sant'Anna School of Advanced Studies in Pisa and the Polytechnic of Lausanne. I deal in particular with neuroengineering, in other words, with recovering the connection between external systems, such as prosthesis, and people's nervous systems, to recreate, for example, tactile, natural sensations even after they've lost a hand.
Dopo aver perso la mano. Il primo progetto su cui
The first project I worked on was GRIP, an Esprit long-term research, which was the father of the EU program FET. Afterwards came Cyberhand and then Neurobotics. I went on to coordinate a FET project called Nebius. Over 15 to 20 years, all these projects have helped us realize a crazy idea, one that could hardly have been funded by traditional channels. This idea has been made available to few patients so far, but they're real patients with real conditions. When we did the first implantation in 2008, the real question was, can we give back sensations to an amputee using electrodes implanted in the peripheral nerves? At that time, we absolutely didn't know. It was a bet.
Non lo sapevamo per nulla, era una scommessa.
Now we've shown we can do it. We've demonstrated that the patient can use this information during real tasks, so it has an impact on everyday life.
Il passo attuale è di rendere questa cosa.
The current step is to make this thing clinically usable for many patients for a long time.
Sicuramente FET ha cambiato la mia vita.
FET has certainly changed my life. It was a training ground where crazy researchers came up with crazy ideas that could be developed without too much fear of failure. This wonderful spirit really helped me. Maybe I could have found other solutions, but surely in this life it has helped me a lot.
Ma sicuramente in questa vita mi ha aiutato moltissimo.
Nanomedicine is getting smarter, improving our ability to treat cancer. The size of nanoparticles makes them ideal vehicles to carry drugs to tumors, and we can modify their surface and material to make them better at moving through the body to reach their targets. Our focus is on treating cancer stem cells, because if we don't get this right, then treatments might not tackle them, and these cancer stem cells will then lead to the recurrence of the tumor.
The goal is to transport therapeutic agents by using nanoparticles as carriers deep into a tumor in order to reach all cancer cells. However, most of those nanoparticles accumulate just at the surface of a tumor, not penetrating deep enough. There are two main reasons for that. The first one is dense extracellular matrix that hinders distribution of nanoparticles. The second reason is abnormal blood and lymphatic vessel architecture. It leads to reduced number of blood vessels, to irregular blood and lymphatic flow, and to high fluid pressure. Taken together, all of those aspects effectively block penetration of nanoparticles. To effectively account for those barriers, we need to very carefully design our nanocarriers. Therefore, in EVA Nano project, we are using agent-based model of tumor growth, where we can very precisely represent spatial distribution and composition of all of those barriers in order to simulate how nanoparticles behave in such heterogeneous environment and how effectively they can diffuse in different scenarios.
We're using computer simulations to test how nanoparticle parameters impact their travel through tumour tissue. This includes things such as the size of the particle or how sticky they are, as well as how quickly they're internalised into the cell. We use a combination of agent-based models to work out how the particles travel through the entire tumour, to stochastic reaction diffusion systems to understand how particles move through subsections of the tissue. Right now we're interested in how space matters, that is, how tightly packed in the cells are, and the material around the cells and the impact of that as the particles move through. We're hoping that these computer simulations accelerates the design framework and allows us to work out which parameters matter the most.
One of the main appeals of gold nanoparticles is that we can attach a wide range of different molecules to them and use it to guide the nanoparticles to the preferred target. These attached molecules create a monolayer coating around the nanoparticle, which influences its behavior. Due to the size and physicochemical properties, functionalized nanoparticles can pass through the biological barriers and enter the cells. And the way by which they do this is really important because ultimately this is what determines their function. This process, however, is highly affected by the size, shape, and surface properties of the nanoparticles. So by understanding how it is affected, we can enhance cellular uptake. In EVA Nano, we perform atomistic molecular dynamics simulations to investigate how the composition of this coating affects the structure in the biological solutions. And then, after obtaining the structure, we can move to a higher level, perform coarse-grained simulations, and investigate how these nanoparticles interact with cell membrane. Results provide guidelines to the more efficient nanoparticle design.
We are dealing with a wide range of scales, from atomistic to whole body scale. Different types of simulations are needed to evaluate at different scales the efficiency of drug delivery by designed nanoparticles. Executing these simulations require a large amount of computational power, but it's not only about how powerful of a computer we have access to. We are looking into finding intelligent solutions to predict the outcome of any given simulations at an earlier stage rather than waiting for the end of the simulations. Our vision is to use explainable AI to understand the intricate inner workings of the interactions of nanoparticles with the environment in which they release the drug they're carrying. We will further use this understanding to evaluate the efficiency of any given nanoparticle design.
We then use artificial evolution to optimize the nanoparticle designs automatically, so we can find new nanomedicine strategies with a click of a button.
By using a simple microfluidic model, we can apply gels that mimic human tissue and then also add nanoparticles that will mimic the nanocarriers of cancer drugs. We can then track these nanocarriers through fluorescent microscopy to gain an understanding of how they might penetrate tissue in the human body.
We are developing artificial tumor microvessel on a chip devices. These devices comprise of microchannel with a diameter in the range of the tumor capillaries and semicircular geometry. Also, they serve as scaffold of human endothelial cells to form an artificial vessel-like endothelium. The devices are used to investigate the transport characteristic of nanoparticle-based anti-cancer drugs in the bloodstream, mimicking as close as possible the human physiological conditions. The microvessel devices are also employed to study how nanoparticles of different sizes or chemistry interact with the vascular endothelium, and how these get extravasated out of the vascular flow to the surrounding tissue into the solid tumors.
The behavior of nanoparticles predicted in silico in EU Nano project are then contrasted experimentally. For this, we are using cancer stem cell model in which the cancer stem cells can be visualized and tracked. We are using 2D conventional cell culture as well as 3D models of the tumor spheres and in vivo models. Importantly, we can visualize and count cancer stem cells before, during and after the treatment. We could as well track its behavior, since we know that tumor as such tend to compensate for the eliminated cancer stem cells in order to survive.
Our ultimate aim is to use our research to help patients. In order to do that, we need to translate our research results into a marketable product. Therefore, we are starting a new project that is financed by European Commission in the program called FET Innovation Launchpad, where we will first position intellectual property that stem from EU Nano in order to develop viable commercialization strategy and reach the market. We will do it through, of course, market research, through research of intellectual property in our domain, and through exploring potential service models, business models, and licensing mechanisms for various stakeholders in both academia and private sector.
I'm Paolo Dario. I hold a degree in mechanical engineering from the University of Pisa, and I'm a lecturer in biomedical robotics at the Sant'Anna School of Advanced Studies in Pisa. I have always believed, and continue to believe, in the FET program, Future and Emerging Technologies. It meant embracing new horizons while setting practical goals in the end, and understanding that applications will be a long way off still. There's 40 years of research behind the bionic hands we have built. We studied worms, we studied eels, lampreys, we studied octopuses. As if we were modern Leonardo da Vinci's, we studied how many organisms work. We didn't copy them, but drew inspiration to create new engineering principles, new design principles, new mechanisms, and new materials. This has been a research focus, one of bio-inspiration and bio-robots. A second focus area is artificial hands and prosthesis. This is also part of what was a long-standing dream, to create hands capable of both dexterity and sensitivity. I'd like to specifically mention the Cyberhand project. Today other projects exist, and my students and colleagues continue working in this area, and they also study artificial touch. The third focus area is a very fundamental project to me, Neurobotics. No one had ever thought about systematic collaboration with non-engineering sectors before. For example, with neuroscience. And actually, this has made it possible to create many different prototypes and to train different people. I'll just mention one of our ideas, a man-controlled capsule able to explore inside the human body, almost as if it was in Fantastic Voyage, the book and the film in which a submarine is reduced to very small dimensions. Then, how to control it with the brain? What kind of interface to use? This is one of the problems we have faced in neurobotics. Or perhaps you may remember Dr. Octopus in Spider-Man, that mad scientist who has a kind of octopus on his shoulders, inserted into his nervous system. Well, can the brain control a body other than its own? So, exploring the unexpected. By the way, this is one of the problems facing Europe. I've often asked myself this. If a 23 or 24-year-old boy like Zuckerberg had come to talk to one of the many top European banks or venture capitalists, they would not have given him a euro. The real challenge for Europe, and for all of us, is to leave the way open to those who are disruptive thinkers, to the movers and to the shakers. And the FIT program was all about this.
This study is an international and multidisciplinary study. It involves partners from several European countries. There are about 40 scientists in this consortium. The main objectives of the EDIT project are to develop a strategy for the early detection of neoplastic lesions. In particular, we want to provide an innovative technological platform for detecting solid tumors below 1 millimeter of size. And at the same time, when we recognize the small lesion, we want to provide a strategy for eradicating the tumor. First, we will try to validate our technological platform on the bladder cancer, because this is the most devastating neoplastic condition. But the main issue in the management of bladder cancer is that there are technological limitations and also therapeutical limitations that means we are not able to detect tumors below 1 millimeter of size. And so for this, the tumors, the small tumors cannot be detected, and so there is relapsing of the tumor. Also, the therapeutical strategies are not effective in about 50% of the patients. So it means that there are a lot of people with bladder cancer that experience relapse after the first diagnosis of cancer. And the relapse can last up to 20 years. It means that the same patients undergo multiple surgical procedures, multiple treatments. EDIT brings together several novel technologies all together in a single technological platform. At first, we want to use gold in the form of nanorods. Why gold? Gold because it is a biocompatible metal, and in the form of nanorods because with this shape the gold can be used for a photoacoustic approach. It means that we can stimulate the gold nanorods with a laser light, and the laser light is placed outside of the body. So upon stimulation with the laser, the gold nanorods will emit ultrasound, so we can detect the presence of gold nanorods through a photoacoustic approach. Obviously, these gold nanorods have to be functionalized to be able to recognize the tumor. So here there is a lot of chemical approaches. So we will bind peptides that are able to bring the gold nanorods only to the neoplastic tissue. Since we talk about bladder cancer, those gold nanorods have to be stable in the presence of urine. So also in this case, we will functionalize the gold nanorods in a way that are stable and can be recognize the tumor in the presence of the urine. We already have very good preliminary results. We published several papers so far, and we also have one patent. Every project is just a preclinical project. It means that we will develop and we will validate all the strategies and technological platforms on preclinical models. The follow-up will be to move to the clinical setup. So the technological platform that will be developed in the EDIT study will change the management of bladder cancer patients, because by being able to detect tumor when they are very small, below 1 millimeter size, and at the same time, if we can eradicate the tumor with thermoablation, so to avoid the issue of chemoresistance, it means we will reduce a lot the relapse of the tumor in the same patient. It means that we will improve the quality of life of the patient, and also while reducing the social cost, because we will reduce the number of surgical interventions and the number of therapies that the patient at this moment goes through. So I hope that this EDIT technological platform will really change the management of bladder cancer patients.
Okay, so thank you for attending. We are here within the Research and Innovation Days session dedicated to Voices from the Future. Voices from the Future is related to the European Innovation Council Pathfinder projects in order to talk about their stories, the people behind the European Innovation Council, and their visions and expectations from the future. Of course, this session is dedicated, as you might already know, is dedicated and is organized within the Future Tech Week, which is a celebration of the European Innovation Council Pathfinder projects. We are going to have these sessions as well as during the afternoon. There is going to be a session dedicated to other relevant stories from the EIC Pathfinder. We are going to have also the interview of the European Innovation Council Program Manager appointed by the European Commission there. So follow us also within the afternoon. And you are able also to see all the results coming from the FET projects and the EIC Pathfinder projects within our future board. So within the Future Tech Week website, you are able to check all the relevant information coming from the Pathfinder ecosystem. So I'm now here. I have the pleasure to open this roundtable session dedicated to the extracellular vesicles research emerging trend. And we are going to talk with a different set of speakers. I'm going to present myself. I'm Marta Calderaro. I'm an Horizon 2020 national contact point for future and emerging technologies, as well as information and communication technologies in Italy. And I have here a different set of speakers coming from the Pathfinder ecosystem. Antonella Bongiovanni from the Best for Us project, in particular from the Italian National Research Council. I have also Maria Goretti Ferreira Siles, which is coming from MindGap project and University of Coimbra. We have Paolo Bergese, which is the coordinator of the EV Foundry project, as well as the BOW project. We have Marcella Chiari coordinating Index, Seppo Vaino, which is the coordinator of the Gladiator FET Open project, and Marina Kretic, which is coordinating the Transition to Innovation Activity project called Marble. So I thank you all for being here to talk about this emerging trend. I would say this research trend that is now on the top of the research and life sciences publication at the moment. So I would like to ask to you, here we have a different set of FET Open projects, Best for Us, MindGap, EV Foundry, but also Index and Gladiator. But before talking about your projects, I would like to understand, and for this I kindly ask and give the word to Paolo Bergese to talk about why the extracellular vesicles are so important today as a research item and should be on the top of the research agenda within the life sciences. Paolo.
Okay, I have to share my screen. Share. Okay, you should see now the screen. That's written extracellular vesicle in a nutshell. So thank you, Marta, for this invitation. I'm happy to be here. And thank you for the others. All of us could have been introduced vesicles in a good way. So it happened to me, and this is my honor. So thank you to all my friends that are sitting here with us for this opportunity. Okay, so let's start from here. Okay, cells communicate, and they can communicate in both a physiological and pathological way. And what was and is written in the books of molecular biology is that they communicate by two ways. So from cell-cell junction and by cell signaling. And cell signaling occurs by biomolecules. So is a molecular way of communicating. But what has emerged recently is that several unnoticed nanolines of communication are now being discovered that the cells use to share information. And among these nanolines, I call them nanolines because these are communication ways that exploit small particles or tubes that have a typical size in the range of the few hundreds of nanometer. And the stars of this communication way are EV nanoparticles that are extracellular vesicles. As you can see from here, we have here two cells, and this is the parent cell, and it can secrete vesicles directly from the extracellular membrane or from this endosomal pathway. But anyhow, the recipient cell is getting this load of information. Here you can see on the right side of the slide, you can see a magnification. So we have a double layer membrane that hosts some proteins, and the membrane is made of phospholipids, and inside you have a cargo that includes proteins and nucleic acids. And the size, the typical size of these vesicles is from, let's say, 50 nanometers to hundreds of nanometers. And this was already observed, these kind of structures were already observed about 75 years ago, but it took a while from basic discovery to arrive to some more conscious understanding and use of these nanoparticles. And so why this was not noticed before? Because it takes a while for the community to understand and keep the right vision on something, and also because these are real nanoparticles. So what helped a lot was the intersection of different point of view. So the point of view of nanotechnology. So you can see in this slide on the bottom level of the slide, you can see the nanotechnology that was synthetically developed in the last 25 years. You can see the gates of the transistor, and on the top you can see some biological nanostructure. And the point is that the connection between these two worlds, different worlds that have been apart for several years, is that they share the same size. So basically the size of a virus, the size of an extracellular vesicles matches the size of the gates of the transistors you have in your computer and laptops and so on. And so when we started to, these two different fields started to talk together to cross-contaminate, started a new consciousness. And well, these are more details about vesicles, but I can skip this. It's just the typical sizes. And the other point is that we are assisting to a sort of debacle of the synthetic nanoparticles. This is a recent study that says that about 100% of the synthetic nanoparticles that are injected, just this small amount, 0.0014% can reach the tumor. So the point is that the nanomedicine based on synthetic nanoparticles seems to be a little bit in a, let's say, it's encountering problems. So the idea is that, and actually, let me go back. So the translation of these nanoparticles is somehow lost, as can be seen here. So the idea is that instead of preparing synthetic particles, we can use and exploit extracellular vesicles that promise to have as you can see in the right side, superior biological and biomedical properties because they are biogenic, they are natural, and these superior properties include best targeting, best circulation, best bioavailability, best and the possibility of personalization and sustainability. So the point is that these extracellular vesicles promise precision nanoparticles for medicine 2.0, so the next generation of nanoparticles. And actually, in the last, I would say, 5 years, maybe a little bit more, but not that much, they have been started to be used as natural, this means cell-free, also nanotherapeutics and vaccines. As you can see, there are some studies, and as you can see on the dates of the published papers and references I put here, it's something that is really happened in the last years. And as you can see in the table on the right side, there are companies that are finally, I don't know if they started with this, with the pandemic going on, but they were foreseeing to start clinical trials in the spring or summer of 2020. Okay.
Paolo. So sorry for interrupting you. So we could say that the information that is gathered within the vesicle could be also used for personalized medicine in a way that we are at the forefront of new approaches within the personalized medicine.
Yeah, of course, these are the personalized or okay, it depends on how you want to use the words, but okay, precision and personalized, if we overlap these two words, it's exactly like that, because they bring inside RNA and genetic material, and so this is one way of being nanoparticles 2.0, but on the other, so they can be also used for drug delivery, so personalized drug delivery, doing proper tweaking and engineering, and they can use as multiplex biomarkers, so because they bring a lot of different biomolecules, and also because of their themselves physical properties, they can be used as biomarkers, but Marcella and Marina will talk about this more, and also Goretti talk about this more in detail. And of course, and this so they can be used for liquid biopsy. And so the idea is that you can spot these particles and get multiple information from disease. So basically, these are the three main foreseen applications. So therapeutics and vaccines, with also some application also for COVID, and drug delivery and diagnostics and liquid biopsy, which is a particular case of diagnostics, and some new perspectives that we will see later on.
Thank you to Mr. Bergese to make a comprehensive overview of what the extracellular vesicles can do for the next approaches in medicine. So I would like to ask you, as well as to, to all the panelists, and in particular, I'll start with Antonella Bongiovanni, to describe the different FET Open projects that has been funded within the European Innovation Council Pathfinder. As we have seen within the different panels, we have 5 FET Open projects that has been funded here. So we have Best for Us. And I will ask to Antonella if she can describe a bit Best for Us project. Thank you, Antonella. We cannot hear Antonella because she...
Antonella, you are muted, sorry.
Thank you. So I just started because I want to start with a feature, one feature of the extracellular vesicle that was exploited in our project, that is also the possibility and capability of extracellular vesicle to act as a cross-kingdom communication system. So extracellular vesicles are not exchanged by cells within the same organisms, but also between organisms belonging to different species. This is, for instance, the case of the microbiota and the human cells. So there are few articles showing that extracellular vesicles are indeed a communication means to crosstalk among species, either for defense or to influence each other. And as Paolo was saying, the other feature that we were exploring in our project is also the possibility to use extracellular vesicle as a novel nanotechnology, biological nanotechnology to deliver drugs for human health indeed. So our project, we named our project FETOPEN. The FETOPEN project started a couple of years ago, best for us, so extracellular vesicles from a natural source for tailor-made nanomaterials. And so we are, the Best for Us consortium is of 6 independent research institutes across Europe, and is a completely real multidisciplinary consortium. So each of us are taking our expertise that is from ecology to chemical engineer or cell biology, and all this expertise is indeed to answer to some limitation that we are facing till now on the use of EVs, the extracellular vesicles for medicine, that are related to the few bioresource from where we can derive extracellular vesicles. And this limitation also is related to the poor scalability of the process giving rise to the production of extracellular vesicle because of the bioresource that is known till now. So briefly, what we proposed in our FETOPEN project in Best for Us, our solution is based on the use of a completely renewable bioresource that is completely, that obviously is photosynthetic. So also for this feature is as an economic value also, since is indeed not necessary to use expensive resource to grow and to have them, is not considered having arable place to grow this bioresource. And so we, the final aim of the Best for Us is in creating a fundamentally new bioprocessing approach that would generate and functionalize extracellular vesicle from this renewable bioresource. So this is.
Thank you, Antonella. I'm sorry, I will ask to each one of you to make a 3 minutes presentation of the project. So I will then pass the word to Mrs. Goretti Sales to present her FETOPEN project.
Hi, first of all, Thank you a lot for being here and for being able to share my experience in science and to get all the opinions from others as well. Our project focuses in EVs, extracellular vesicles. Of course, that's why I'm here. But the panorama around it is completely different. So it binds basically the knowledge that there is a communication between the body and the brain. And this communication is established because the brain-blood barrier allows some messengers to pass over. There are many things crossing, including EVs, and we have the assumption that these EVs are bringing on communication. One important pillar of this communication is the effect of the mind in this communication. And this brings up the subject like if you practice meditation, it has been clear in the literature now that you will be healthier, your health indicators improve, and it is a science fact, it's no longer some common sense. And on the other hand, it's also a science fact that if you have stress and you are worried with stress, you will get sick. And the question is, how is this interrelation between the brain communication with the body and how the mind affects it? If there is any relation, this should be by some information that is being transmitted through the brain towards the body. So basically, what we need then to study in this Mind Gap Fat Open project is to close the gap between this body and brain and mind interactions that we don't have a clue right now what happens. So we are looking for the messengers of the mind. What messengers can our brain send to the body when you have a mind-controlled operation? And we have several approaches to have some kind of control, if that is possible, but we all know, and it's common sense, that mindfulness will bring you some kind of mind control. So we are going to have a set of hundreds of patients enrolled in this process, so that they have a mind control event, and we are collecting their blood and saliva and fluids so that we are able to monitor then the vesicles that they are sending through the body, vesicles that are from brain origin, and we are checking their cargo. So we need to build up a machine that is able to monitor that. Overall, we need to gather different disciplines, and interdisciplinary is very important, and this allows us to gather several top researchers in the world working in specific differentiated fields that meet together in this Fat Open in a very convenient way. So we need people that handle EVs specifically, and that is SEPO that is also there. We are going to grab the EVs that come from the brain with plastic antibodies, and so we need experts with plastic antibodies. We also need informatics because EVs are a tricky species, so we need some help of the informatic people, and we are going to make this device in a microfluidic setup. In the end, of course, we need some clinical sciences altogether to this main-brain relation so that we end up in the end with a conclusion that is valuable for people. So these are the partners of this project. Is Coimbra University, where we are coordinating, but we also have Olo University, that's from which SEPO comes. We handle IPO Porto Hospital that will handle a clinical trial that is already recorded, and it is expected to involve more than 700 people, expecting that COVID does not bring additional news in the middle. And we have VTT to do the microfluidics and Linnaeus University to plan all these informatic things. So we are bringing up innovation in two main branches, and this is what I want to highlight from MindGap, is that we are bringing a new knowledge in this relation between body, mind, and health, which has been some kind of diffused and no science has permeated into it. And I think it is the first time that we are going to have a science-based evidence that there is some messengers that are being transported from the brain so that the body gets health when you control the way your mind works. And of course,
thank you to Mrs. Sales. I'm sorry, I will just close with this information regarding that the mind can drive information also through vesicles, to have extracellular vesicles. And I will pass the word to Mrs. Marcella Chiari, which is the coordinator of the Index Fat Open project. Thank you, Professoressa Chiari.
Okay. Thank you very much for the invitation to this roundtable. I have I summarize in this slide what is the aim of INDEX project, which is already clearly explained in the title, by the way. So the key words of these projects are extraction, detection, quantification, characterization of extracellular vesicle present in body fluids. Because if one wants to use vesicle, extracellular vesicle in diagnostics, which is the aim of many of the research activity going on in these days, one has to consider that in the blood there are many, many nanoparticles similar to extracellular vesicle. And if we analyze and collect all of them, it's a big mess. We are not going to understand anything. So basically my idea was, if we want to really have a tool that can be transferred to diagnostics, we need an instrument, basically, a platform, an instrument that does two things that are described in this slide. We need a platform where the extracellular vesicles are collected from blood, and then we need a module that analyzes them. I knew two groups, one in Paris and one at Boston University. The group in Paris has developed this technology based on magnetic particles, and I suggested them to apply their technology, microfluidic based technology, to the separation of extracellular vesicles. The Boston group was using interferometry to analyze virus, and viruses are very similar to extracellular vesicles, the same size, the same similar behavior in a way. And so I decided to create a consortium with these two groups. And the problem was, if we collect extracellular vesicle using magnetic particles, so basically we bind them to magnetic particles, then we have to find a way to release them intact so that we can analyze them individually on the surface of a chip. And for this, our group found a solution that is based on DNA-directed immobilization. Basically, we use DNA linkers to capture the extracellular vesicles, and then we release the vesicles by cutting this linker. And this idea turned out to be successful, and we filed a patent based on this idea. And this patent, as Marina Kretic will say, is the basis of the new project. When we started, this concept of analyzing intact particles was really new. Because the interferometric technology at the basis of this detection module was not so popular, I would say. But since we started, a startup company originated from Boston University, NanoView, they collected a huge amount of money, and they are going into this idea of analyzing the particles by interferometry. However, our project is different in the sense that we integrate not only the analysis, but also the collection, which they don't have. And basically, I'm happy that the NanoView has become successful, because this demonstrates that actually there was really a need for this application. And I'm convinced that INDEX project can do even better and advance the field even more.
So a need in terms of market and also interest from possible venture capitalists to find.
Absolutely, because they collected millions based on this idea, actually.
Okay, I will then pass the word, I'm sorry, Professor Chiari, I'll pass the word to another FET Open project, which is coordinated by Seppo Baigno, which is the Gladiator FET Open project. Unfortunately, you are mute. Seppo, unfortunately, you are mute. We cannot hear you because...
Here, so now I guess it's better. Can you hear now? Can you hear me now?
Yes, yes, we can hear you.
Excellent. It's a pleasure. So I'm representing the Gladiator program that is under the FET Open, and this illustrates a complex setting where we actually use the EVs or the exosomes as a sort of nano communication tool in the body that then aims to use their kind of unique property, what Coretti was just mentioning also, and Marina earlier, that basically these EVs are known to cross the biological barriers, and one of the critical barriers, the blood-brain barrier, that has really hindered scientists and medical doctors to really kind of establish very effective therapies that would target the brain. So if you actually then want to treat diseases like brain glioma, which is one of the severe brain tumors, so then there are drawbacks that this blood-brain barrier is preventing this. So this Gladiator program really brings expertise of simulation of the nanocommunication properties that we get, sort of vesicles that would be then targeting the brain with respect to the glioma. And then it builds on our discovery that basically the EVs are able to be responsive for radiation frequencies. So basically then cells are kind of based on the radiation, pushing out distinct types of qualities of EVs, and then we can use this as a novel innovation to actually develop medical application called radiogenomics, which then couples the exosomes that are then kind of secreted from these kind of machines, kind of, or organoids that are actually engineered to express specific vesicles that then are kind of regulated externally from the body, and then these are targeting to the brain. So this consortium brings capabilities to make organoids, which is represented by the Fraunhofer capabilities, and then we have also the computing capabilities that are dealing with the radio technology from the ENTU from Norway, and then we have also properties of the Greece and Cyprus that are dealing with the oncogenesis of the brain, and also developing the reception systems like the antennas that are needed to couple. So this is kind of the actual program is represented in next, which is the ultimate goal that we actually want to achieve novel therapies that are then providing kind of long wanted and needed help for the people that have brain gliomas. And then this is sort of illustrating the system where you have a transmitter that is actually the actual cells that are responsible for the radio frequencies that are then secreting specific EVs that are then secreted and diffuse and go across the blood-brain barrier to reach the actual target to treat the disease.
Thank you.
Yes.
Thank you, Professor Vaino. I'm sorry, I'm just make a brief introduction to all the projects, but I missed one FET Open project from Professor Bergese, which is actually supporting as well extracellular vesicles. So I asked to Professor Bergese, which at the moment is mute, to describe EV Foundry and then describe also the BOW project. You are mute, Paolo. I cannot hear, unfortunately, Paolo.
Sorry, I found right now the. Okay, now can you hear me now?
Yes.
Yes.
Okay. Yeah, I have to apologize. First, maybe you see myself moving, and I'm not doing something else, but because I have two screens and I'm trying to manage all the presentations and all the stuff. So, but thank you. Okay, so this is the EV Foundry project, and the basic idea of us was that, well, if we want to play with vesicles and produce things, and we want really develop vesicle nanotechnology, we need to get vesicles from some sustainable sources. So to find a way to have abundance and reliable samples of extracellular vesicles. So the idea is depicted here. Our sustainable sources are milk, bovine milk, and the culture media from Ascaris, which are those parasites that are hosted by pigs. And please understand that these two kinds of vesicles are very interesting for humans, because we are used to eat those vesicles since, and we are educated to them since the very beginning of our life. Here, what's moving, you see, we are using microfluidics. You can see a branch of vesicles, and you can see that they can be separated. We use DLD, so DLD nanotechnology. And what I wanted to say, and this is an important thing about FED projects, that you can give the possibility to young researchers to be hired and to work on the project. On EV Foundry, we hired these very beautiful researchers, 10 of them, plus of course a farmer and a butcher for the pigs and for the bovines, and is a project that is a participant about the Italian consortium of big interfaces, the University of Utrecht, Aarhus University, Lund, ANSA Biomed, which is a small company, and the University of Southampton. And well, and the nice thing is that we are off the way and we have already good results. So the thing is that basic science space and these FET projects can be very, very useful for for laying down the basis for research, because a lot of basic research can be done, and this set the basis for future development.
And actually, I take the word from what you have just said regarding the impacts on the EV Foundry to ask you about the FET Proactive project, BO, that was actually a continuation of EV Foundry as well as Best for Us project.
Okay, yeah. I switched presentation. So, do you see the screen? Okay, so actually the FET, so EV Foundry was a FET Open, and thanks to the cluster events and to the connection that we started to make between FET project, this is the BO project is one of the first things that came out from there. Actually, the BO project, this is what I was saying at the beginning among us. So many basic research can drive one innovation. So you need more basic research for one innovation. And actually, the BO project comes from three European projects. The MADIA project, that is about magnetic diagnostic assays for neurodegenerative disease, and this is an ICT collaboration project, the EV Foundry project, and the Best for Us project. All these three projects come together in the BO, that is biogenic organotropic wetsuits. The idea is to put a wetsuit made of the extracellular vesicle membrane, so is to dress up the magnetic nano devices developed in MADIA. So basically, you can see here the scheme of the particle. Inside, this is not a sick vesicle, but instead is a vesicle that has been emptied of its biological content, and now you have this magnetic diagnostic assay. But to make it acceptable, let's say, to the immune system, the idea is to cover, to wear on it the suit made of the vesicles, and so let's say the membranes and the vesicles, they come from the Best for Us project, while the dressing cabin, the dressing technology comes from EV Foundry and also from Best for Us, so they come together. And so this is how this comes out, this idea. So to provide the nano devices with a wetsuit that masks them from the immune system and gives them the availability, the property to target specific organs, to cross blood-brain barriers, for example, and so on. So this is the idea. This project is bigger and longer. Actually, it is a 4-year project that will be kicked off the 1st of November. And we have a big budget, and actually we are 11 partners that there are really 16, because some partners then have different departments participating in the project. And is this big because we start from the production of the vesicles, the synthesis of the nanoparticles, we put them together, and we have a very, very extended plan for in vivo testing of the performances of these nanoparticles. This is the reason why we have such a big group.
Thank you, Professor Bergese. I would like to, talking about results coming from FAT open projects, in this case, EV Foundry Invest for Us, and also Madia project has given the possibility to support the BO idea, related idea.
No, they gave the possibility, the materials, the concept, and the connection between the people. So they are really made the BO project possible at several levels, not just from the technical and technological point of view, because it emerged from discussion with Antonella.
This is exactly also the way that was in a way creating an additional opportunity from the index project, which was presented by Professor Chiari, for the creation and the gathering inputs for the Marvel project, which I would like to ask to Professor Marina Kretic to present to us.
Thank you, Marta. I hope you can see my screen now in presentation more. Sorry, I don't want to. Okay. So thank you, Marta, for this great initiative. I'm very, very happy to be with you and share our opinions on this topic. So Marvel was funded within the frame of the EIC Transition to Innovation Action, a specific call dedicated to take a result from a FAT open project and bringing these results to a increased technology readiness level, up to a level where we can start talking about commercialization of these results. And so Marcella has already described you this result of INDEX, which is this reversible DNA-directed immobilization of antibodies used for the reversible isolation of vesicles. And these results of INDEX that was actually patented provide a technology that provide us the possibility to obtain vesicles with high purity, with high recovery yield, with high selectivity, but was developed for a microanalytical scale, for the microchip that Marcella showed you before. The aim of Marvel, the ambition of Marvel is to produce a technology that is scalable. The technology developed within INDEX is not scalable due to the presence of antibodies that, as you know, are complex bioreagents, expensive, with sometimes problem of stability, of batch-to-batch variability. And the Marvel leverages on this technology and specifically on the DNA-directed immobilization, but proposes a paradigm shift in the use of the capturing molecules, a paradigm shift from antibodies to peptides, to be used in place of antibodies. Why peptides? Because they are completely synthetic molecules, so they are scalable and they are quite cheap, they are stable, they can be easily manipulated and modified. And the ambition of Marvel is to apply this peptide-based technology coupled to different kind of cleavable linkers, to different solid supports, to create a versatile platform that can be applied to different scales. So from the analytical scales of microliters in the lateral flow devices, for example, that are tiny analytical devices, to preparatory lab scale, for example, for the analysis of urine, when we talk about milliliters, up to the development of system of isolation for manufacturing scale of extracellular vesicles, where we talk about liters of culture media. And so, and Marvel actually will be validated, will be demonstrated in two important relevant environments, clinical environments, that are the regenerative medicine, and specifically cardiac repair by the production of therapeutic EVs from cardiac progenitor cells to be used in post-infarction recovery, and also in the liquid biopsy, so the diagnosis of bladder cancer by the analysis of vesicles from urine, so a totally non-invasive method to diagnose bladder cancer. So the impacts of Marvel can be described broadly in precision medicine, because we aim to have better clinical outcomes at lower cost, improve life expectancy, improve quality of life, sustainable public health, and so on. But moreover, what I'd like to underline here today is that in line with the transition to innovation activity called philosophy or vision, Marvel really wants to take the index technology or results of index technology towards commercialization. So really want to bring it to a real usage. And to do this, basically, we have designed this draft exploitation path that I'd like to show you today, because it's a clear explanation of what it's the philosophy of this granting scheme. So starting from a concept developed within a FAT Open project, an exploratory project that developed some different results. One of these results was taken at TRL3, and thanks to Marvel will be hopefully brought towards a technology readiness level of 6, where we will start talking about the creation of a business, the drafting of a business plan for the creation of a startup able to commercialize the platform developed by Marvel. Of course, for doing that, two things are very important to me. A real contamination of different actors of research within Marvel, because we need the researcher, the basic researcher, the academia, the research institutes, but we need the end users, so the clinicians, and we need the industrial partners to make this step from TRL3 to TRL6. So this is very important. And the other things is that, of course, we will need in the future more funding for sure from public and private, and from the public point of view, we will for sure look at the other EIC funding schemes that are the SME dedicated instruments and the SME instrument and the FTIE schemes.
So thank you to Professor Kretic. This is actually the real hidden value of the European Innovation Council, and the way the European Innovation Council expect to support breakthrough innovation. But of course, as we have seen today with this list of different projects that has been funded, this is a research-driven innovation, and this is something that we need to highlight in a very relevant way. So actually, talking about this, about the different impacts, Professor Bergese was starting to say it in a way, mention the clustering initiative that was supporting also all the different projects to sharing experience. And I would like to ask to Mrs. Goretti Sales her experience and her expectation and also willingness that you have find within this clustering initiative that you 5 projects have created some years ago. You are mute, unfortunately.
Sorry, Martha. This cluster idea was indeed an idea of Antonella, Marcella, and Paolo that found by coincidence that there were several FAT Open projects integrating the idea of exploring DVs for disruptive research. So when they found it, it would be interesting to cluster so that we could cross-fertilize our knowledge because then we can move further. And it's interesting because I have been given the possibility to have also cross-collaborative effects with some of the projects here. And this is bringing up into the field some events as well. We started this clustering. It was hosted by Antonella from Vesperas. And we started in Palermo last year on the 6th November. And we were supposed to do now in Coimbra this year, Portugal, but unfortunately the COVID changed the ideas, but we are going to establish a remote a virtual meeting as this one, but later on, it's not scheduled already, but we have the compromise to have a near meeting to share our data all together among all the FETOpen projects, and now Proactive as well, that are joined in this idea of exploring DV technology. So, and most of all, and also, we also have a manifesto that was made public to emphasize the importance of DVs on the community and to show up very clearly that there is today some restrictions in technology that need to be handled by research, and only research-driven evaluation or scientific growth can allow us to bring out the EVs into the market, knowing obviously that EVs can produce many changes in the way we see many fields, from food to health to agriculture. So it is important that we move further away EVs, and this clustering event was really a great opportunity.
This is thank you, Mrs. Sales. This is actually something that is pretty relevant and important, because now the European Innovation Council is talking about the creation of these projects portfolios. So this is exactly an example of what a project portfolio is. And I would like to pass the word also to Mrs. Bongiovanni, Professor Bongiovanni, regarding the Italian EVITA association that was created after the identification of all these different projects coordinated by, in that specific case, Italian coordinators. But we can see that there are not only Italians here, of course. Antonella.
Yes, thank you, Marta. So indeed, actually, the Italian Society for Extracellular Vesicle, named EVITA, was founded in 2018, and indeed was just after meeting Marcella and Marina. So actually the FETOpen that we were coordinating was key in also giving us this idea of building up a new society. And this is the result of the growing interest in the extracellular vesicles, because we are not the only European society on extracellular vesicles. Actually, there are, I think, more than 25 society on these topics in Europe and all over the world. And again, so we were having last year the first meeting, and we were almost 200 scientists, Italian scientists working on extracellular vesicles. And I would like now to launch also our new idea on doing this workshop on EV-based biomarkers, that is going to be, unfortunately, again for the COVID-19 pandemics, is going to be remote. So it's going to be virtual in September 25th, and it's going to be one day, and it's going to be open for all European scientists working on EVs, free workshop for people interested on. And so eventually I want to just show the home page, so if people be interested on it, it can follow us on the EVITA Society webpage. Can you see the screen? So,
yes,
and all the information are there. And this is again a very important point, because bringing together scientists is really key in advancing science and innovation. And this is, I mean, we can tell just for the experience of us that is indeed the case. Okay.
And this is actually something that it's quite important, because you have talked about more than 20 national association in your specific research community. So what about creating, all of you, a European association about extracellular vesicles? I mean, this is something that maybe could create the basis, you know, for making this priority more strong within the European policy itself, and as well as the research and innovation community around Europe and at the international level. So now I would like to ask to Professor Chiari about the main specific characteristics that you find relevant for the creation of this kind of projects, research-driven innovation. We have talked about risky research. So what do you think about this?
Well, I think that the funding scheme for breakthrough technologies must be maintained. It's extremely important that there is a specific scheme for breakthrough technologies. And actually, FET-Open is the only funding scheme for high-risk, high-gain collaborative projects in new technologies. And we must consider that if we are prompt to build projects that are only based on TRL already high, the more risky ideas are lost, and sometimes risky ideas fail, but in many cases the risky ideas or risky projects generates a strong knowledge, which is not necessarily transformed immediately into innovation. Maybe it becomes innovation some years later, after it has been shared among different research groups. So I think it's extremely important to support projects that have where the TRL required is from 1 to 3. And this is not always understood even by the reviewers of these projects. Sometimes the reviewers ask, what is which TRL do you have? And they want us to be more close to something that is innovation. But these projects are based on knowledge development. That's very important. Another thing that needs to be absolutely there is the interdisciplinarity and the multidisciplinarity, because it's essential to accelerate the transformation of the idea into something that can be more close to innovation. So I think that they cannot stop there, by the way. We need another scheme, another funding scheme to, let's say, develop further what is the knowledge that is and the innovation that is generated within these FET-Open projects.
Thank you. We have seen also from Seppo and Goretti slides from both of them about the relevance of interdisciplinary consortia here. So within a collaborative perspective to grow the research idea to something that can be, I would say, innovation, but maybe cannot. So Goretti, what is your feeling about this?
I think interdisciplinary is very important, because FET-Open is a well-designed program in that aspect. It allows you to have, and I fully agree with Marcella, a radical view over something. And it is important to know that society is looking for radical views and for changes that promote us further away. And that is only possible with radical ideas that can be plausible within a short time. And that is only feasible if you have interdisciplinary events. And FET-Open is well designed because only partners with specific fields of knowledge can gather. If they are superimposed, your evaluation won't be great. So people just cluster together if they have different areas of knowledge. That means that something new is going to arrive there, and that gives the possibility to cross-fertilize different knowledges into a unique product that will arrive to the market much closer than, much faster than, for instance, if you have an ERC grant, which is a blue sky idea in which you are most likely all alone. I had an ERC grant previously. I was very happy to have my idea fulfilled within 5 years, but it is a very risky position. But indeed, when you go together with different people working in different fields, you can go much faster in science. So I hope all these schematics, the RC, the FET-Open and FET-Proactive, are able to maintain within time, because they are very important, because they are like the main pillars of the knowledge-based society that we are looking for.
Thank you, Goretti. I would like to make a question also to Paolo, in particular regarding the FET-Proactive. I mean, what are your which are the differences from a FET-Open and a FET-Proactive project from your perspective, and which should be the differences from a FET-Open and FET-Proactive project? You are mute.
Well, the main difference is that the proposal is the double of the pages. I mean,
no, this shouldn't be the difference.
Well, it's a big difference when you prepare it. Okay, now I'm kidding.
No, can I ask something about this? Can I ask something about this? Yeah. Which one you prefer?
The short version.
Well, I prefer the shorter one.
Okay.
Yeah. I prefer, because it's 30 pages. It's a lot. You have to go a lot through the details. So it's hard, because then you risk to become too much descriptive, and reviewers don't like that.
I'll ask the same information to Seppo afterwards, but please continue.
Well, I think no. Okay, I think that the thing is that the point. I think that the idea is, as already Marina showed up, the idea to continue a project. But what I found in the code that may be implemented is that we succeeded in communicating to the reviewers and to the community our idea. But the fact that you put together different projects in the background of your idea, this is not really considered and written in the code, because the code is more straightforward, as Marina showed very nicely. So so it was harder to mix different projects, because this is something that was not considered in the code. And I mean, the frame, the template did not consider this kind of let's say
possibility or
next step, right? So I think that. And also about the TLR, because the TLR means that you start from an idea and you go on. Instead, we brought on 3 different projects, so it is a new idea coming from 3 different projects, but the TLR is still low. Okay, so we took forward what was done before, but not in the linear way of development as the project Marvel. So it's this kind of framework was not set very well set in the template and in the code. So we found hard time to do that.
What do you think about the information of the call for proposal related to the community building? This is actually something that has been done with by your clustering initiative regarding create a community around this topic. So, but what do you think in general? Do you think that this is something that is an added value for a FET-Proactive topic or not?
Well,
yes,
I mean, talking with people is always useful, but yeah, I actually Maybe someone else has a better
Okay.
answer than mine.
Can I say something?
Because we are really growing up as a team. So this is a question for Antonella.
Okay. I'll then pass the word to Antonella.
And the Evita Society, they are good for that.
So what I think is that the scientists, many of us are individualists, so they like that much to work by themselves and not sharing, not that happy to share. So, and I think that is not just by chance that this cluster is made by us from the fact. So I don't think that there are so many clusters at the ERC, because I think that the fact structure of the project force you or select people that love and like to work together. So the multidisciplinary and the fact that the group, so it's not a single entity applying for the FACT project, but you need to apply as a team, as a group with different expertise. This is absolutely another value, because allow you to get used to work together and enjoying the working together and exchange and sharing experience and expertise. And so it's not by chance that we end up so we started from the FACT, the single coordinator with the FACT, and then we end up with this cluster that eventually will put some other maybe FACT projects working on EVs. And then me and Paolo ended up having a novel project together, and maybe with Goretti is going to be some other kind of collaboration. And this is, as Goretti was saying, this is a key. I mean, this is the real science that is made not just by yourself in your lab, talking with with a very small community, but opening up.
Also, as Antonella said before, also the FAT Open Art project, one of the reasons why Evita was born, the Italian Society was born. So.
Can I ask, because this is the real science Antonella was saying, and actually I would like to ask about the real innovation, since Marina was taking the word. I mean, you are coordinating one of the latest funded project in the pilot transition to innovation activity. And this is something that it's quite relevant also to understand. We have talked about interdisciplinary collaboration, talking about, of course, disciplines, but what about academia versus enterprises versus consumers versus final users and users of the technology?
I believe that is definitely contamination between academia, pure research, and users, for example, clinicians and industrial partners, is fundamental to build really a product, to move on the innovation chain. There is the innovation chain does not advance without the basic research, but also as a researcher, I daily learn something working with the end users and working with industrial partners, and the other way around, of course. So this contamination and interdisciplinarity and multidisciplinarity is absolutely fundamental.
This is quite good, and
for the advancement of this innovation chain, really.
Of course, and this is actually something that we have seen also within the European Innovation Council new calls, because of course we had the FET Open, the FET Proactive previously, but we didn't have the FET Innovation Launch, but the transition to innovation activities, which are supporting the innovation chain, the innovation path. So I would like to thank you all and asking for a final remark. So I will start from Seppo about your willingness for the next European Innovation Council Pathfinder scheme. What you would like to see there?
I mean, basically that we are promoting, first of all, this kind of innovation chain value building, basically that starts from the basic research and then goes really for this building of innovation, so that this kind of primary discoveries are developed then for actually end users. And this is something that we have to nourish. So it's like this primary basic research is like a seed, and then we kind of put that into the ground, and then we need to nourish that in the European sort of to be innovative and really create totally new things that makes us competitive for global scale. So that's more or less, I guess, the message for the, the politicians that keep us alive and, support the ground and the seed research, which is the basic research that then leads to new things. And this EV research is really exciting, that it has given and will give us exciting openings, as we've heard today. So that's a good choice for funding this, and the best for us is a great initiative, and I really like it to bring.
Thank you. Thank you so much. I will ask to Antonella.
Yes, just briefly. So I'm completely convinced that the innovation breakthrough is taking place when there is a very close link between research and science and market and companies and SMEs. And when this link is very close, as the FACT program was fostering and suggesting and pushing on us to do it, for sure is going to be, the innovation will take place. So I will not, and I really hope that either in the EIC a vision where the science perspective will come together with the market-oriented innovation. So this is my
Thank you.
idea.
Marcella? You are mute, Marcella.
Sorry. I want to go back to Brussels. Sorry. I want to go back to Brussels. I want a lot of money, investments, and meet in person the other researchers, be able to submit many projects in a simple way. One of the things I would like to suggest is that maybe we can have a different approach to evaluation. So maybe we can have a more interactive approach to evaluation. This is something that sooner or later we will have to consider, because when the projects are really innovative, it's very difficult to find a multidisciplinary. It's very difficult to find a panel of evaluators that cover all the area in detail. So maybe a good idea would be a preliminary evaluation of the projects, and then to be maybe invited there and discuss with the panel in person, be able to answer all the questions directly, something like that.
This is what
Point taken.
Yes, and I has this, yes.
Point taken. Thank you, Marcella. Goretti, a 1 minute final remark. I'm sorry, I have to.
I want to full support the idea of innovation, radical vision that is necessary to have a knowledge-driven society, as all want. And this requires, of course, funding, and funding in different TRL levels. If the next funding schemes keep as they are excellent, because they allow us, and we are privileged persons, to be able to have money to drive our research further on. We like to have radical ideas, and sometimes it's difficult to put them working if we don't have money. So FET Open is very well as it is. I would recommend that it stays as it is, unless it brings some additional improvements. But the funding scheme is brilliant, and I think it's a very important opportunity for society, so that they invest in research, and research can give back the money with innovation. But by gathering people that really know the knowledge, and those people are in the universities, and combining them with the industries that will help drive that to the market. So that confluence, that joint point must exist. We cannot separate areas, and we cannot let some loose points. We need to put people working together to be able to drive the products into the market.
Okay, then thank you, Goretti, and the last word to Marina and Paolo. First, Marina.
Okay, yes, I definitely agree with Goretti that this FAT Open, and with the other speakers, that the FAT Open funding scheme is brilliant. And also I liked very much the fact that some of these results can be sustained to an increased TRL leveled by different schemes of funding with progressively increasing the TRL. So we can, as Marvel demonstrates, really start from a FAT Open and then go on, and this is really great. As a final, really brief remark, I'd like more bottom-up approach and less top-down specific challenges to follow, and that's all.
Thank you, Marina. And then, so more bottom-up and less top-down approaches. And then the latest word to Paolo Bergese.
Thank you. Well, okay, I agree with all my friends that talked about this before, so I have to say something different. So I will say to the Euro, look, with extracellular vesicles, we provided the first example of biogenic nanotechnology. Means a sort of nanotechnology 2.0 that's covering from the fundamental science up to the innovation. There's a lot of collaboration going on. There are societies. So Europe, I think we're ready for a flagship-like initiative about extracellular vesicles. That was not in place anymore, but I really think that extracellular vesicles can drive research and innovation on the new era of nanotechnology that is bio-compatible and sustainable. Yeah, we're ready for the fact.
So, thank you, Paolo. This was actually quite good because you have supported the team, the research team of all of you. So as a final remark, and this was actually quite good. So I would like to thank you all for your presentation and also for your different information that you give to us about extracellular vesicles. And thank you all for being here within the Future Tech Week. Let's see each other within the Future Tech Week and the Future Board. Thank you. Bye-bye.
Thank you. Bye.
Bye.
Hi, and welcome to Marvel. Marvel is a project funded by the European Commission within the Horizon 2020 FAT Pro Act program. Marvel partners are from 5 European countries. The project is coordinated by CNR, the National Research Council of Italy. There are 2 clinical partners, Cardiocentro Ticino from Switzerland and Università Vita e Salute from Italy, and 3 SMEs, Hyperdrop Diagnostics from Spain, Hansa Biomed from Estonia, and Amires from Czech Republic. Marvel mission is to develop the first affinity-based, scalable and reversible isolation technique for small extracellular vesicles, opening entirely new scenarios in both therapy and diagnostics. Indeed, Marvel technology will be validated in 2 important clinical contexts that are the diagnosis of bladder cancer by liquid biopsy and cardiac regeneration by cell-free therapy based on extracellular vesicles. EVs are an heterogeneous family of submicron membrane vesicles released by most cells in basically any biological fluids, with a fundamental role in cell-to-cell communication. Much interest is flourishing towards their exploitation for innovative methods in medicine. To date, the highest clinical relevance is thought to reside in exosomes, nanovesicles smaller than 200 nanometers.
Exosomes represent the third generation cell product to be used in a cardioprotective and cardioregenerative medicine. After the initial excitement over cell transplantation using adult progenitor cells for its relative easy to use and good safety profile, the initial mechanistic hypothesis has been questioned. To date, we know that cells have been effective by indirect mechanisms, by reducing apoptosis in cardiomyocytes, stimulating angiogenesis, or modulating the inflammatory reaction. Thus, the trophic secreted molecules and secreted vesicles are more critical than the differentiation of cells in the new cardiomyocytes. Protocol to isolate in a large scale the well-defined and most effective fraction of exosomes, as it is for one of the Marvel project milestone, is of paramount importance to move on toward the clinical application of cell-free approach in cardioprotection.
On the other hand, extracellular vesicles have a role in pathological processes as well. Secreted by malignant cells, they have been implicated in disease development and progression by stimulating tumor cell proliferation, angiogenesis, extracellular matrix degradation, and immune response deregulation. Since one of the most important challenges about cancer is the diagnosis at early stages and proper treatment, the identification of biomarkers represents an urgent clinical need. Resembling malignant cells of origin, tumor-derived extracellular vesicles are suitable surrogates to be considered as a source of diagnostic biomarkers. Upon isolation for liquid biopsies, the analysis contributes to implement non-invasive, affordable monitoring of cancer progression.
Marvel key technology can be declined in one simple word: peptides. Our vision is indeed to introduce a paradigm shift in the affinity capturing of extracellular vesicles and use synthetic peptides to replace antibodies.
Peptides' properties can indeed be finely tailored in terms of selectivity and affinity of EV recognition. We will indeed develop different peptide probes acting both as general EV binders by targeting EV membranes and peptides targeting specific EV protein markers to enrich predefined EV subpopulations. We will take advantage of the versatility of peptides as synthetic molecules to enable their conjugation to different supports. Conveniently, we will also design and apply different strategies to enable the release of extracellular vesicles under mild condition, preserving their integrity. We will then exploit these concepts in the integration of our affinity systems across different settings and operating scales, including LFTs, laboratory kits, and TFF membranes.
In Marvel, the breakthrough is to develop an affinity membrane unit for the large-scale purification of extracellular vesicles by using highly selective hybrid probes specific for subpopulation of the vesicles. Currently, for the isolation of vesicles is applied sequential filtration by integrating different membrane processes, microfiltration, ultrafiltration, diafiltration. But one of the drawbacks of the site-based isolation is the presence of nanoparticles with the same size of the vesicles and the presence of contaminants molecules. In Marvel, we will develop a hybrid of modified membrane unit to be integrated with the tangential force protection system that currently is used by enhancing efficiency and reducing contaminants.
Over the last few decades, EVs have become sort of magical bullets that fueled high expectations for new generation diagnostics, omics, and therapy. Hansa Biomed has pursued this long-term vision since 2007, becoming a global pioneer in providing EV-focused solutions to life science market. While today, along with Marvel Consortium, we're looking upon other markets such as that of precision diagnostics and regenerative medicine, which impact reaches millions, billions of euros in high-interest hubs such as Europe and States, and can bring benefit to hundreds of millions of people worldwide. We also consider other markets such as nutraceuticals, cosmetics, and consumer care as early release markets that are even more lucrative with less regulatory hurdles and shorter product development cycles.
For paper-door diagnostics, as a rapid diagnostic test startup, Marvel will provide novel and disruptive know-how that will be applied in new products and future collaborations. From our experience, European projects have always helped to increase the synergy between research groups, industry, and healthcare systems. Technologically based startups can benefit from European innovation programs to improve the technology transfer flow from research and industry to the European healthcare system. Thanks to the funding obtained, startups can hire highly trained personnel, such as PhDs, and explore cutting-edge and multidisciplinary projects.
In 2020, EV Therapeuticals have entered human testing all the way to COVID-19 therapeutical trials, but developers still face some challenges regarding the EV exceeding heterogeneity and complexity that impact GMP, regulatory QC issues. So within Marvel, we want to reshape the state of art and advance it versus truly affordable, scalable, robust solutions that we will then launch either within existing pipelines or we will establish strategic partnerships with therapy developers or dedicated startups. In any way, we will try to attract new public and private funding.
Marvel will address different target groups, including research community, industry, SMEs, end users, broad public, policymakers, and European Commission. The website and social media will be one of the main windows into all our activities and results. We plan to directly engage the broad public not only through articles, press releases, non-scientific publications that we plan to publish within the Marvel project, but also through organization of different events that we plan to organize over the next 2 years. And finally, Marvel project will also engage into certain clustering events with the participation of policymakers and European Commission, where we will present all the possibilities of the Marvel project to those target groups.